Now look at this.
What's up about penguins? Today we're going to do topic 1.5, which is on the structure and function of biological macromolecules. So let's first talk about nucleic acids. So nucleic acids are going to be made up of nucleotides. Nucleotides are made up of a pentose sugar that's going to have a phosphate group attached to it as well as a nitrogenous base. Nitrogenous bases can be one of 5 different things. You could have a pyrimidine or purine. The pyrimidines are going to be cytosine, uracil, and thymine, and our purines are going to be adenine or guanine. Whenever we have bonding that takes place within DNA, it's always going to be a purine and a pyrimidine. So adenine pairs with thymine, cytosine pairs with guanine. A couple things to note about that. Adenine and thymine are going to have 2 hydrogen bonds, and cytosine and guanine have 3 hydrogen bonds. Now notice, because of the fact that we have a purine and a pyrimidine paired together, it always has an equidistance. It's kind of in a way of like 3 ring structures across. So if a purine is replaced by another purine, it's not going to affect the width of the DNA molecule. But if a purine replaces the pyrimidine in the DNA structure, that's going to cause potential issues due to the distance or the width of the DNA not being the same. So with our DNA, you're going to notice that the strands are going to be anti-parallel. So that means that they're going to be equidistant apart, as we said before, and they're also going to be running in opposite directions. So there's a 5' end where your phosphate group is, and there's a 3' end where the hydroxyl group is. So let's switch gears and talk about proteins for a moment. Proteins are going to self-fold. And so our primary structure is just going to be our string of amino acids. It'll be held together by peptide bonds between our amino acids. Our secondary structure has to do with hydrogen bonds between the backbone. Between that polypeptide backbone, there's hydrogen bonds, which will lead us to our secondary structure, which where we'll see alpha helixes and beta pleated sheets. Our tertiary structure has to do with the final 3-dimensional structure. This is the R groups interacting between the different amino acids. So that's where we get our final 3-dimensional structure. And the quaternary structure is going to be the R groups interacting between different polypeptides. So if we look at this tripeptide, we're going to notice that there's 2 sides to it, right? There's the N terminus, which is where the amine group is, and then there's a C terminus where the carboxyl group is. This is the directionality that's important to know for proteins, just like the directionality that we saw with our DNA with the 5' and 3' ends. Let's talk about lipids for a second. So there's 3 different lipids. You have fats, you have phospholipids, and you have steroids. Fats are made of a glycerol and 3 fatty acids. And as you notice here, the phospholipid still has 2 fatty acids, but we replaced one of the fatty acids with a phosphate group. And steroids are made up of 4 fused rings. So you see here in cholesterol and cortisol, we have that 4 fused ring structure that makes up our steroids. If you have penguins, just success.